So I missed the email on Shakespeare. And I realised as I was sitting there that you just can't fake Shakespeare, so I'm not even going to try. But it was a real privilege to follow, Seth. Thanks very much. You probably know that I'm filling in for Mark McCamish, who was my predecessor. He retired 2 months ago. And while he's kicking back in California, I've got the unenviable task of filling in his huge shoes because he was a pioneer at Sandoz. He led the development of the 2 out of the 4 US approved biosimilars. And I'm also giving his presentation, so I hope I do him justice. I realised this morning also that the presentation does cover very similar ground to the presentations this morning, but this is probably not unexpected because we're still working in a really new area. We're working with relatively limited published data, and I do think it's still worthwhile to look at the same issues from different perspectives. And the reason being that in any new area, it's the way that academia, industry and regulators learn from each other. And I think in this important area, it's absolutely essential. I know talking with some of the colleagues this morning that not everyone here is familiar with biosimilar development. So apologies that some of this presentation will be motherhood and apple pies to some, but hopefully it'll be useful for those less familiar. I often get asked, I spent the first 8 years at Novartis working on the originator side, and I've just recently joined Biosimilars, and I get asked a question a lot, working for a company where we both do originator development and biosimilar development, how I found moving into the biosimilar space. And I say the same thing, which is that it's the enthusiasm and commitment of people who are involved in developing biosimilars is infectious. And the reason it's infectious is, as Seth was saying, is that they really see this as a mission. And the reason it's a mission is because the pharmaceutical industry has a social compact. And that compact is in return for exclusivity and essentially a monopoly on the originator product, the way as an industry we give back is to make that innovation available for patients. And so for my colleagues at Sandoz, their mission is all about access to medicine. The other thing I say is that unmet medical need can be addressed by innovation, and we can drive the boundaries in the practice of medicine by developing drugs which are better than anything we've seen previously, and that's where innovation is bending the practice of medicine. But unmet medical need isn't just addressed by innovation, it's also addressed by access. And in my previous life, when I was practicing infectious diseases in extremely resource-constrained environments in countries in Africa, I had the experience of having to manage patients where I just couldn't access the drugs that we take for granted. And when you see what happens when patients can't access drugs that we take for granted and we know work, it's not a good situation. And so this is why we feel very strongly that biosimilar drugs are essential, because it's a way of fulfilling, I think, industry's compact with society, and also because it's about addressing unmet need through improving access. So what I'm going to talk about today is, I've touched on why biosimilars are important. I'm also going to talk more about the concept of biosimilar development. The word Janet is coming back again, extrapolation. I'm sorry about that. I've also got a couple of pyramids in my talk, so I'll show you the pyramids as well. And I'll touch on post-approval requirements and then just conclude. So I've mentioned that biosimilar drugs are very important, and that's because the role of biologic drugs in medicine has dramatically changed the landscape for many patients. They have a critical role in the care of patients with renal, hematologic disease, rheumatology conditions, as well as in cancer. And in fact, in many guidelines in these diseases, biologic drugs are recommended for use in patients. And it's really revolutionised, as I said, the practice of medicine in some areas. For example, in psoriasis, before the anti-TNFs became available, it was a very difficult disease to treat. It involved either the application of very inconvenient topical applications or being exposed to UV irradiation. And really the new agents, the anti-TNFs and the new agents, are inducing clear or almost clear skin in the majority of patients, and some of the new agents also inducing remission. And this has a massive effect on people's lives. Now, we've heard a lot this morning that the manufacturing and the analytical basis of biosimilarity underpins the endeavour. And being a biological system, the process is complicated, and there is going to be natural variability in the drug product and drug substance. But this can be well controlled during the manufacturing process. And we very much agree with the analytical basis of biosimilarity, obviously, and not the least being because physicians and patients expect a biosimilar drug to be high quality with proven efficacy and safety, and that's going to be a consistent product regardless of who made it. Now, the high cost of biologics are creating treatment access issues around the world. And this, I think, chart shows some of the rationale for this. In dermatology, for example, costs are an important barrier in about a fifth of patients. And this is in European countries and in Canada. In European countries, about 1/4 of the 46 members of the EU cannot provide access to any biologics for the treatment of rheumatoid arthritis. In the US, about 1 in 4 cancer patients will delay or omit treatment due to financial considerations. And we've heard a lot about reimbursement this morning and the complexities there. In rheumatoid arthritis, patients who are eligible for third-line treatment in RA, only about 76% of patients or 65% in Germany, 66% in the UK can access. And this is why biosimilar drugs are important for people, because limited access may result in some patients who may be more severely affected being treated when disability or morbidity or complications can either be prevented or limited with earlier treatment. And Sandoz is very much about ensuring access for patients who would otherwise benefit from biologic drugs. So high quality, low cost biosimilar drugs equals improved access, and that is the mission that motivates colleagues who work on this. So what is a biosimilar? Well, in the top panel is the US regulatory position. It's a biological product that is highly similar to the reference product, notwithstanding any minor differences in clinically inactive components, but there must be no clinically meaningful differences between them. In Europe, it's a similar statement that an approved biosimilar medicine and its reference contain essentially the same active ingredient and are expected to have the same safety and efficacy profile. And obviously these are regulated by a stringent regulatory process in both the EU and the US. And from a patient's perspective, these statements of the regulators are extremely important because they can expect the same safety, quality, and efficacy from a biosimilar product. If there's any doubt about the outcome, then sponsors should not seek approval of a product and regulators should not approve the product. So here's the pyramid again. It is actually a useful concept because we've heard this morning, I think Seth mentioned it very well, that the understanding of biosimilar drugs is not quite there, and we have a different mindset in physicians. And I think this slide explains why. Obviously, on the left, with originator drugs, the major goal is to establish the clinical safety and efficacy. And there's less attention or less weighting put onto the analytics and non-clinical data. Obviously, it's a progressive development. But the difference with biosimilar development is that the major goal is to determine biosimilarity by establishing a scientific bridge to the clinical experience of the originator. And the analytical methods to do this provide the most sensitive tools to establish the scientific bridge. And during the development of a biosimilar drug, all the experiments and studies are designed to establish biosimilarity, which sounds like an obvious statement. So in development, the first step is to really analyse the originator to understand the biophysical attributes of the product. And this defines the goalposts for the manufacturing process. I'm going to show you some data shortly that although this seems like a straightforward endeavour, it isn't necessarily straightforward. And I think Dr. Woodcock, used the words startled. I think as you look more closely at reference products, you do discover differences that I think maybe even the originator manufacturer wasn't aware of. The next step is to, in an iterative process, and I think you saw a chart this morning, the black box of the manufacturing, really need to deconvolute that, and that takes an iterative process focusing on things like the cell line development, the bioprocess development, protein purification, and the drug product development. And in this iterative process, which can be more complicated or less complicated depending on what you find, is the basis for establishing the analytical bridge and the consistency of the manufacturing process. And the goal here is to optimize the process so that essentially the biosimilar product matches the originator. And finally, this will provide the analytical basis for biosimilarity, which then can be confirmed in non-clinical and clinical studies. Now, the process can be controlled at different levels, and just briefly can be controlled at the stage of raw material, in the process design, in the in-process testing and the control of the process parameters, and also in the release testing of the harvest, the drug substance, and the final dosage form. And all these steps are important, obviously, in ensuring overall control of variability of the manufacturing process and also the consistency of the product. And obviously, these controls are mandated by various guidelines, regulation, and laws. Now, how similar are biosimilars and their reference products in biochemical structure? Well, clearly the amino acid sequence, the primary sequence, needs to be identical. In fact, even if one of the 1,300 amino acids in a monoclonal antibody is different, the FDA will not approve that, and I think Dr. Woodcock said that very well. Folding needs to be the same to elicit the function, and the structure-function relationship is obviously crucial. And minor differences in glycosylation and byproducts from the biosynthesis and post-translational modifications are allowed only if the clinical relevance of these differences can be shown not to be relevant. And in terms of the clinical program, clinical testing is relatively insensitive, and it's just to confirm biosimilarity. Now, in terms of the analytics, these have changed, and that's been alluded to this morning, over the last 20 years or so. And just to put this into context, if you look, for example, at mass spectrometry, in 1990, the technology allowed one to determine a 100 picomolar difference in an analyte in a glass of water, essentially. These days, you can detect a 100 picomolar difference in an analyte in an Olympic-sized swimming pool, which represents an increase in sensitivity about 10 million times. So you can imagine that, I think as Dr. Woodcock said this morning, when you apply these increasingly sensitive tools to reference products and indeed biosimilars, it becomes clear, where the goalposts are, what you need to do. And I think this is producing biosimilar drugs that are becoming ever more close to the reference product, which is a good thing, obviously. Now, whilst biosimilars are designed to match the variability of the reference product, it's also clear that reference products change over time, and that's been described this morning very well. And non-identicality is a normal principle, obviously, in glycosylated proteins because it's produced in a biological system. But no batch of any biopharmaceutical is identical to other batches. And this natural variability isn't necessarily a problem, even in the human body, and that's reflected in the guidance. Now, manufacturing changes are made frequently, and differences in attributes can be greater than batch-to-batch variation. And these manufacturing changes are stringently controlled and approved only if they do not lead to clinically meaningful differences. And I think Paul made the point that not all manufacturing changes lead to changes in the molecule, which is very true. On the right side shows some data published in 2013 with rituximab, showing on the top panel the concentration of fucosylated moieties and varying batch to batch as described by the expiry date. If you look below that, you'll see that there was a manufacturing change around 2010, which resulted in a shift in the ADCC potency. And you can see the shift in the dark plots, and on the right in unfucosylated sugar moieties as well. So variability, and in this case, for example, this wasn't put through clinical testing. The changes were deemed to be not clinically relevant, and so this was allowed. So variability per se is not the issue, but control within the acceptable range is the goal. Now, I mentioned that manufacturing processes can change, and this is a chart showing the frequency of changes. You can see, for example, infliximab's had more than 35 manufacturing changes, compared to canakinumab, which has had about 1. And the changes are well understood. They can just be methodological, they can be more fundamental, such as change in line or purification methods, but these are tightly controlled by the regulators. The interesting thing is that biosimilars became possible when regulators became comfortable with regulating changes in manufacturing. And if you look at this chart, you'll see that FDA guidance was issued in the mid-1990s, and this turned into the ICH Q5E guideline in around 2004. And this was about the same time in Europe when the EU law was changed to allow development of biosimilars in around 2004, and this was followed about 5 years later in the US. So when health authorities were confident regulating manufacturing changes, this concept was extended to biosimilars and is underpinning the framework that we see today. So as mentioned, the goal of clinical development is not to prove de novo safety and efficacy of the biosimilar. It's really to confirm biosimilarity. And here are the steps. Now, you would think that the cycle time should be shorter because the burden of evidence is less because of the bridge to the reference product. It isn't always shorter. Sometimes it's as long or longer depending on what problems you have. And I think both for industry and regulators, it has been a learning process where we've learnt the variability isn't always as controllable as we'd like. But the goal of clinical development is to establish biosimilarity, not to establish safety and efficacy. So from the point of a clinical program, these need to be sensitive enough to detect differences, and they may include different study design, different populations compared to the original studies with the reference product. And herein probably lies some of the uncertainty in the prescriber community about biosimilars. I mean, people are used to looking at controlled studies published in major journals establishing the safety and efficacy of originator products. And this isn't true for biosimilars, and people often want to see the data. And this really leads us to, again, extrapolation and the fundamental thought. And the idea here is that the same molecule will behave the same way as the original molecules in all indications and patient populations. And when we talk about extrapolation, it's important to say what it is and what it isn't. So why is extrapolation possible? Well, it's possible because it's based on demonstrating a level of sameness between the reference product and the biosimilar product. By demonstrating this sameness and ensuring that any differences observed are not clinically relevant, then extrapolation is possible. So if there are differences in sequencing or glycosylation, for example, that result in functional differences that are measurable by bioassays, then extrapolation is not possible because the analytical bridge hasn't been established. It can't be established by clinical data alone for the reasons already outlined. And what it is not is extrapolating from one indication to another. And we know this because not all drugs are approved in all diseases, and it's not necessarily true that if a originator drug is not an approved indication, we would not seek to extend the indication. And this forms the basis, obviously, of having extrapolation only in the approved indications. So extrapolation is not about extending clinical data beyond the reference product. This can be illustrated with this published example. It came from our group in the manufacturing changes of Enbrel that were approved. The analytical data with the molecule revealed manufacturing changes that weren't previously reported, and these changes were evaluated and deemed to be highly similar or comparable. And the modified process was approved as producing a highly similar product, and therefore its use was extrapolated to all approved indications. And this is despite the fact there was a reduction in the concentration of one of the sugar moieties from 50% down to about 30%, but no functional difference, no clinical relevance. So the health authority approved the change without any changes to the label or any other changes in the use of the drug. Having said that, extrapolation isn't that well understood by the medical community, again, because all this data is not really visible. Physicians don't see the data, it's not readily published, the basis of biosimilarity is not as accessible as data from originated drugs. And this is one of the challenges, I think, of getting biosimilars established. I think Seth said it very well. It needs to be something which is supported by patients, by manufacturers, by agencies. And so this is the totality of evidence you've heard about. So matching structural attributes, biological functions, human PK/PD, and confirming biosimilarity in a clinical study using a sensitive population in a sensitive indication means that other indications can be justified. So what about following approval? And we've heard something of what FDA expects on post-approval safety requirements. And for any biological product, obviously post-marketing surveillance and pharmacovigilance applies equally to biosimilars as it does to originated products. They're expected to have the same safety profile, but over time, particularly with less common events, it's possible that for infrequent events, the safety profile will converge and diverge over time as more data is added to the data set. And obviously, good pharmacovigilance is important for all biological products. I think also, as has been talked about this morning, the role of real-world evidence in things like switching and things like understanding immunogenicity and understanding or generating data in populations which are extending the clinical data beyond the approved indication are going to be important, certainly in some payer contexts. I'll just skip that slide because it talks about pharmacovigilance, but certainly at Sandoz, our pharmacovigilance procedures are identical with originators. So just in conclusion, a biosimilar and its reference product are expected to be the same. The regulations allow this. It's confirmed by more than 10 years of experience with biosimilar products in EU. Obviously, all products require the same level of pharmacovigilance, and I think other optional post-approval activities such as non-interventional studies and registries certainly are going to provide absolutely important and useful data to address scientific questions both of payers, prescribers and patients moving forward. And I think, again, Seth said it very well, it's all about confidence of patients, prescribers and regulators. So thanks very much.